In this session, I'm going to begin with a new chapter, body fluids and circulation. And under this chapter, we'll see about the circulatory system in humans. The circulatory system in humans, it comprises blood, then it also has the blood vessels. So we know the circulatory system in humans is a closed type of circulatory system, and then we also have a pumping organ called heart. So this is a pumping organ. The blood vessels will include arteries, arterioles, capillaries, venules, and veins. So these are all the blood vessels, which we'll study in the coming sessions. So first we shall see about this blood. So this blood is a special connective tissue, and this blood has mainly two components. So one, it is plasma, and another one, all those cells will be together called the formed elements, right? So this plasma will be constituting around 55%, and whereas these formed elements will be having, altogether it will be 45%. And coming to this, right, plasma. So this plasma is mainly composed of water. So the water will constitute around 90 to 92%, right? And then it also has proteins that constitute around 6 to 8%. Okay, it also has minerals, then it also has clotting factors, has glucose, amino acids, then lipids, etc. So the plasma, it contains water, proteins, minerals, clotting factors, glucose, amino acids, lipids, etc. So coming to these proteins that are around 6 to 8%. So these the major proteins in the plasma, it will include like a fibrinogen, which is an inactive, right, clotting factor. Then the next one, globulins, like immunoglobulins, and then albumins. Okay, right. So these are the major proteins. So these fibrinogens, right, are the one which help in coagulation of blood. So help in coagulation of blood. So which means the clotting of blood whenever there is an injury. And here the globulins are involved in defense mechanisms. They are involved in defense mechanisms, right? Whereas the albumins will help in osmotic, so help in maintaining osmotic balance, right? So that's the water balance. So here fibrinogen help in coagulation of blood, and globulins are involved in defense mechanisms, and albumins will help in maintaining this osmotic balance. Again, coming to these minerals. Okay, so what these what are all those minerals? We'll see some minerals, okay, present in the plasma. I'll mention here. So those minerals which are present, it will include, okay, sodium, right, and calcium and magnesium consists of, okay, and then bicarbonates are also present, and right, the chlorides. So these are all the minerals which are present in the plasma. Okay, so sodium, calcium, magnesium, bicarbonates and chlorides are the minerals present in plasma. So these are the components of plasma: water, proteins, minerals, clotting factors, which we'll see in the coming sessions. Okay, glucose, amino acids and lipids. The plasma without the clotting factors. So I say plasma minus, okay, clotting factors. If you remove the clotting factors, okay, from the plasma, then you will get the serum. Okay, so plasma minus clotting factors is serum, right? So here the serum contains antibodies. So it contains antibodies. Now let us see about those blood corpuscles or the formed elements. So as I already mentioned that the blood is composed of, okay, two components, plasma and another one called the blood corpuscles or the formed elements. Now we'll see about those, okay, formed elements. Now these mainly, okay, are RBCs, so red blood cells. So in short we write the RBCs, right, which we call them erythrocytes. Then next we have white blood cells, in short the WBCs, right, and these are called leukocytes. And then the third one are the blood platelets. So these blood platelets are called thrombocytes. So they are called thrombocytes. So here the normal values of RBC, WBC and platelets, it will be in the case of red blood cells, it is 5 to, right, 5.5 million RBCs per cubic mm. Okay, and coming to white blood cells, the value is around, okay, 6,000 to 8,000 per cubic mm, right? And then in the case of thrombocytes, the value is around 1,50,000 to 3,50,000 per cubic mm. So per cubic millimeter, so you will find in the case of RBCs 5 to 5.5 million, whereas in the case of white blood cells you will find around 6,000 to 8,000, whereas the blood platelets, which we also call them thrombocytes, are around 1,50,000 to 3,50,000 per cubic mm, right? Now these white blood cells, we have further types. These WBC cells, we classify them into granulocytes and agranulocytes. So depending on the presence of granules in their, okay, in those cells, if granules are present, then we call them granulocytes. If they are not present, they are called agranulocytes, right? So these granulocytes are also called, okay, polymorphonuclear leukocytes. So these are called polymorphonuclear leukocytes. So in short, I will write, okay, as PMNL. So that is polymorphonuclear leukocytes. So these polymorphonuclear leukocytes include, so it includes the acidophils or eosinophils. So these acidophils or eosinophils, they comprise around 2 to 3% of those, okay, total WBC cells. Then we have is basophils. So these basophils are around 0.5% to 1%. Okay, then the third one are neutrophils. We'll see about them in the coming session. So these they comprise around 60 to 65% of the total, okay, WBC. So the granulocytes, which we call them polymorphonuclear leukocytes, because the shape of the nucleus is not circular, it shows different, okay, shapes, and basing on the shape of the nucleus, so these cells will be identified. So the acidophils or eosinophils, then basophils and neutrophils. So among these three, the least one will be the basophils. Yeah. Then coming to these agranulocytes. So these agranulocytes, so it consists of again two more types. We call, okay, one type as these lymphocytes, and another one are monocytes. So these lymphocytes will be constituting around 20 to 25%. The lymphocytes are around 20 to 25%, whereas the monocytes are around 6 to 8%. Then these lymphocytes are again further, we have two more types. Okay, so one we call them, right, the right, the B lymphocytes and T lymphocytes. So there are B lymphocytes and T lymphocytes. Okay, so I will write it, okay, freshly. So I said these A granulocytes, which I have classified into, okay, lymphocytes and monocytes. Okay, right. So these agranulocytes are lymphocytes and monocytes, and these lymphocytes that constitute 20 to 25%, and these are around 6 to 8% that just now we have seen about them. Okay, and I told that these lymphocytes are again of two types. One I call them, okay, T lymphocytes, basing on, okay, where they actually mature. If the usually lymphocytes are all developed in the bone marrow, so if they reach to the thymus where they get mature, then those lymphocytes we call them T lymphocytes. And if they mature within the bone marrow itself, we call them the B lymphocytes. So T lymphocytes and the B lymphocytes. So these T lymphocytes, so where the T stands for the thymus. So in this case, the B stands for, okay, bone marrow. Of course, in case of birds, they have right, birds are fabricaceous. Okay, in case of birds, okay, so they have, so the birds are fabricaceous. So birds are, okay, fabricaceous is the one where right, the lymphocytes will undergo maturation. So I'll write, so where lymphocytes undergo maturation. Okay, in our case we don't have bursa fabricaceous, we have only the bone marrow. So in the bone marrow, if the lymphocytes get mature, then we call them B lymphocytes. Now we'll see these T lymphocytes. We have further, okay, types. We have T helper cells. So these T helper cells, in short I call TH cells. So these are also called as the CD4 cells. So here the CD stands for cluster differentiation. So CD stands for, so cluster differentiation. Right, so here this cluster differentiation actually refers to cell surface proteins. So there are some, okay, proteins present on the surface of the cell, so which means they are in bound with the cell membrane. So we have is here cell membrane, and there are some special proteins that are in, okay, embedded with this cell membrane and projecting, okay, to outside. So these are what we call them, okay, they have given some numbering and all. Basing on that, they will identify what type of cell it is. So basing on that, okay, so this is a right CD4 cell. So this CD4, which stands for the cluster differentiation, and it is, I'll mention the CD4 is a co-receptor. So the receptors are again will be seen like this, projecting, okay, from the cell membrane, and this CD4 is a co-receptor for the class MHC, okay, 2, for the class, okay, MHC2, and it is present on T cells. Okay, so of course the MHC stands for major histocompatibility. I'm sorry, right here, so it's a major histocompatibility complex. So the MHC stands for major histocompatibility complex. So this CD4 is a co-receptor for that class. Okay, so in the coming sessions we'll see that what actually these major histocompatibility complex molecules, and so it's a co-receptor, okay, for class, I'll write here, class 2 MHC molecules on T cells. Then the next type will be including, like, the T C cells. So I call them C, okay, T cytolytic cells. So T cytolytic cells, these are also called TC cells, right, or also called CD8 cells. So just like here in this case the T helper cells, right, as I said that they are co-receptors for class 2 MHC molecules on T cells, these CD8 are also in the same way. So the CD8 stands for, of course, that is actually a co-receptor for, okay. So in this case, this is a molecule for class 1, MHC molecule. The CD8, okay, the CD8, that is cluster differentiation 8, is a co-receptor for class 1 MHC molecule. Okay, and this is also, we'll see on the T cells. So hence, okay, so you can also call these TC cells as CD8 cells or the CD4. Okay, I mean, in the case of T helper cells, as the CD4 cells. Right. The next one, the next type of T helper cells, they include, okay, T suppressor cells. So these cells will help in suppressing the activity of okay, immune system. So after their, okay, defense mechanisms, these in short we call them as the TS cells. And again, one more we have is the T memory cells. So in short, I call them TM cells. These T memory cells, okay, are the one, right, which will remember about that microorganism that entered, or the parasite that entered and caused the disease. It will remember about, okay, those right parasites, and it can help in triggering, right, the secondary immune response. Right. So the next one, we'll see about these monocytes. The monocytes, they include the macrophages. So they form the macrophages. So these macrophages I will be classifying into two types. One, I call them as wandering macrophages, and another one as fixed macrophages. So if those macrophages are only present in the tissues, we call them, okay, fixed macrophages. If they are, okay, moving all around the body, we call them, okay, wandering macrophages. So these one example of these fixed macrophages, it include like uh, the Kupffer cells in liver. So the Kupffer cells in liver are fixed macrophages. Okay, right. So that's about a brief idea of all those, okay, components of blood. So here, all these, okay, blood cells are formed from a, right, single mother cell. What we call it as, okay, the pluripotent. So pluripotent, okay, hematopoietic stem cells. So it's called pluripotent, okay, hematopoietic, right, stem cell. In short, I write it as the P, okay, HSC, the pluripotent hematopoietic, okay, stem cells. Now this is the actual mother cell from which all blood corpuscles are formed. Right? The next one, the pH of blood. So the pH of the blood is about 7.4. Okay, and here the plasma proteins act as acid base buffers. So they act as acid base buffers, and that they help in, okay, and thus they help in maintaining pH of the blood. So the pH of the blood is 7.4, and okay, so the plasma proteins act as these acid base buffers and help in maintaining the pH of the blood. So now we'll see here right, one by one all those blood corpuscles. So first in that case we'll see about the red blood cells. So the first one are BCs. So that are erythrocytes. So now this, okay, right, the formation of RBCs is what we call hemopoiesis. So the formation of RBCs is called hemopoiesis, and the formation of RBCs, okay, occur in, okay, yolk sac. Okay, so in the order I am giving you, okay, yolk sac and liver. So this you will see during the, okay, developmental stages. So during the development of the embryo, okay, then fetus. So you may see, okay, in the first, in the instance you will see the RBC production in the yolk sac, then liver, and then finally in the case of it occurs in red bone marrow. So it takes place in red bone marrow, okay. So which will be the component of long bones, okay, in the ribs you will see this red bone marrow. So that will be the location of formation of RBCs, okay, right. Then the lifespan is around 120 days. The lifespan is 120 days, and these, okay, mature RBCs do not have nucleus. So the mature RBCs do not have nucleus, but okay, the those RBCs which formed, okay, they will be having nucleus, but during the maturation process the RBCs will lose, okay, completely the nucleus. So you can say that in this case we say erythroblast. So this erythroblast will have nucleus, but later once it becomes erythrocyte, so it loses the nucleus. So this erythroblast is, it has nucleus. So whereas erythrocyte do not have nucleus. So it's a mature, okay, RBC. So we call it erythrocyte or red blood cell here, okay. Then so these RBCs contain hemoglobin. So it's a right respiratory gases carrier like carbon dioxide and I mean oxygen. So RBCs carry this oxygen and of course the carbon dioxide. So even hemoglobin will be carrying. So all the how I think you have seen in the previous session, so how this hemoglobin will help in carrying this oxygen and carbon dioxide, and so right. So RBCs contain hemoglobin, okay, right. And so this a healthy person. So in a healthy person, the quantity of hemoglobin is around 12 to 16 grams per 100 ml. So it will be around 12 to 16, okay, grams per every 100 ml. Then decrease in hemoglobin content. So I'll write here. So decrease in hemoglobin content is a condition we call it anemia. called anemia. Then, right, increase in RBC count in RBC count. So the increase in the RBC count is a condition what we call it erythrocytosis, called erythrocytosis, or also you can call it okay, polycythemia. Right, so then, and another one, the decrease in okay RBC count. Okay, so decrease in RBC count, okay, is a condition we call it erythrocytopenia. So it's called erythrocytopenia. Then the total percentage of, right, so I'll mention here like, so the percentage of RBC occupied in total blood volume is what we call the hematocrit value. So it is called, I'll write here, so it is called the hematocrit value. So it's a hematocrit. Then the ratio of RBC to WBC, so it will be around okay 700 is to 1. So if I say the ratio between RBC to WBC, it will be for every 700 RBCs around 1 WBC will be present here. Okay, then for the maturation of RBCs, okay, so certain vitamins are required. So it requires vitamin B12 and also the folic acid. So these two are important for the maturation of RBCs. If they are not present, then it results in low count of RBC. That condition we call it polycythemia. So here with this I'll stop here. So in the next session we will see about okay the different types of WBC cells, and also we'll see about the blood vessels, their transverse sections, and so right.